Hello and welcome to our Webinar Wednesday lineup. I'm Anja Röben, Product Manager for Cell Biology and Molecular Biology at BioSign, with more than 20 years of experience. We are very proud that so many people, customers and curious scientific workers join our webinar today. Thank you very much. The aim of BioSign webinars is to transport new and exciting information about laboratory technologies and new products to all interested people. Today, we will share comprehensive information with you about 3D cell culture with InoMatrix, a new and exciting material for biomimicking cell culture in 3 dimensions. I'm very happy that Ivana Krupova and Eva Proseka from Inocure today present us a better insight into this exciting theme. Ivana is product manager for InoMatrix cell culture plates at Inocure, and Eva is working there in the research and development department as senior scientist. At the end of the session, we will have about 5 minutes for discussion. Please type your questions via chat into the system whenever you like. We will answer them at the end. Thanks in advance for your input. Ivana, please go ahead.
So, hello everybody. I hope you are doing well despite the weird and strange circumstances as COVID pandemic is, but I'm happy that we are here and we can talk about the science and about the very progressive method, which is 3D cell culture. So thank you BioSign for inviting us and having us. Me, as a product manager, I would like to just shortly and briefly introduce Inocure company, and then my colleague Eva, which has more than 15 years of experience with the cell culture systems, will be much more concrete and detailed in 3D cell cultures, and she will be talking and giving you the tricks and tips about our product, Ino Matrix. So Inocure is a nanotechnology-focused company. We focus on nanotechnology for medicine and biomedical and medical technologies. So just for briefly informate you what we do. So we have several technologies. The first one is InoMatrix and 3D cell cultures, which we will be talking later about that. And then it's important to mention that we are also a producer and manufacturer of electrospinning and electrospraying devices. Why it is important? Because InoMatrix is making of nanofibers. So just for briefly inform you how this works. So on the picture you can see the fibers. This is our lab desktop device which we launched last year. So you can see electrospinning method live right now, and the nanofibers are coming from the emitter to the collector. Then we launched also in this year big industrial line. It was like an answer for the fighting with COVID system. So this is the big industrial line where you can see how the nanofibers are making in a big picture. So we are producing face masks, filters for air filtration against viruses. But now I know that you are really interested in 3D cell culture. So let's talk more about InoMatrix. So thank you one more time for having us. And now I will give my word to the expert, cell culture expert, Eva Proseka.
Thank you, Ivana, and good morning, gentlemen. And I would like for your time today and the opportunity to introduce to you InoMatrix, the 3D matrix for 3D cell culture. InoMatrix is a 3D fibrous membrane which can be used for 3D cell culture or for organotypic models or as a system for drug delivery systems. Membranes for 3D cell culture. Why to use 3D? How you can imagine human body that is not flat system, right? That is not 2D. We are 3D architecture. So therefore, 3D cell culture is also necessary to imitate the natural environment for cells. And moreover, fibers, nanofibers, are imitating even the natural fibers in the extracellular matrix. However, how you can imagine, each tissue, each organ in the human body has different morphology or different properties. So we are also able to get with InoMatrix close to these parameters by adjusting chemistry of the fibers or their physiological properties. Currently, we prepare the 3D InoMatrix membranes or scaffolds in four main morphologies, either in microfibers or nanofibers, which are much smaller, how you can see, much thinner and more dense, or their combinations, or porous fibers. And each of them have some properties suitable for one type of cells or the other. How here you can see that we already tested our membranes with many types of cells, for example, human mesenchymal stem cells, where you can see wonderful communication between the cells on the 3D matrix. Also, some cancer cell lines, adipose tissue cells, or recently pluripotent stem cells derived hepatocytes. We are nowadays able to also develop and set the best properties suitable for your needs, for your cells. So we are able to influence the fibers or porosity, fiber size, roughness of the fibers. You can imagine that it's a different scaffold for bone tissue, which is very hard, or for example, liver tissue. That's also regarding to alignment of the fibers, either not oriented or oriented fibers. We are even able to blend some inorganic additives such as hydroxyapatites or blends with natural polymers. Very exciting field is core shell fibers, where we are able even to encapsulate bioactive molecules inside of the fibers and influence their sustainability or their release with protected activity of the substances. We also able to make different transparency of the scaffolds, more suitable for high throughput and microscopy, that you can image your cells without flipping of the non-transparent thicker membrane, which is more suitable for smaller experiments, smaller batches of experiments. For drug encapsulation are many systems, so we really always find the best approach to protect the substance or meet the needs for your systems. So here only a few examples how by the, let's say, playing with the chemistry and physics of the manufacturing of the fibers, we can influence if the substance will be released like burst release, really fast release in same actually time point, or if there will be more sustained release within a time range. So now more about the things, matrix, how to start with the 3D cell culture. We offer a morphology kit, a selection plate, which contains all these four mentioned of the fibers, either nanofibers, microfibers, or their combination, and porous fibers, which are also suitable more like absorption of the bioactives or substances from the gross media. Your cells, you even can try coatings which you used to use for 2D cell culture. You can compare it if it's even needed, because the cells are attaching very well to the fiber morphology. So you can try your let's say protocols which you used to use for the 2D cell culture, and then usually like pilot tests, MTS tests, to see how the cells proliferate and are viable on the groups of the membranes. And then you can select the most suitable membrane for your cells. How I already mentioned, you can use protocols and methods which you used to use for 2D cells, either spectrophotometric methods or microscopy. Recommend confocal microscopy if it's possible, if it's available at your department, because then you can nicely see how the cells ingrowth into the membrane actually. And for example, we were able to go even up to 200 micrometers of the depth with confocal microscope. You can of course use also more methods which are highly recommended to confirm your results, such as qPCR, ELISA, Bioplex, Luminex, each current methods. I would like to share with you a few results where we compared the 2D cell culture with 3D. For example, with the cell adhesion, you can see the cells adhered very well first day after seeding on the all InoMatrix membranes. Comparing to the 2D cell surface, here you can see the cells communicated. Really, you can see that it's flat, like only like until the tight junction, only within the 2D surface. Unlike the Ino Matrix membranes, you can see the cells are within different levels and communicating within the 3D architecture. Also, another test of cytotoxicity or cell viability, 3D. Here you could say that, oh, 2D monolayer is much better, right? It's within 2 weeks, within 14 days, like first day even more proliferated, 7th day, and even more 14th day. And here we can see on the day 7 a drop of the proliferation. However, day 14, again, the cells started to proliferate. Here we can also see the differences between the InoMatrix membranes, because here this test was performed on pluripotent stem cells derived hepatocytes, which are smaller cells which needs really tight communications and be really close to each other, so they do not prefer much the big pores, which actually the porous fibers have. we can see what happened or what we observed by other method, because purpose of this test was to see differentiation of the pluripotent stem cells into hepatocytes, and if the 3D environment can develop differentiation of the cells. And here we can see also the reason for the drop of the proliferation on the day 7th and day 14th, comparing to the 2D monolayer, because were proliferating very well but not differentiating. And comparing to the InoMatrix, we could see that the cells were differentiating by that time and showed high levels of metabolic markers of hepatitis. It was very nice also results. Oh, sorry, it was too fast. Also to perform more assays from one membrane, because all biologists know how much to perform experiment with significantly and statistically valuable results when, for example, the spectrophotometrical assays we need to use. Yeah, it's very nice to have at least group for all the molecular biology methods, at least 3 samples. Yeah, the more is the better. So now we tried to actually save the material and also the cell culture material, the meat. So far 4 assays from one sample. So this figure is from a sample on which we performed LDH assay from the conditioned medium. Then we add nanoglow assay, washed the sample and performed MTS, washed the sample and cells with fluorescent staining for cell adhesion. And you can stayed nicely attached, and we could observe nice cell adhesion on the membrane. That is also another advantage of 3D scaffolds or membranes, that the cells stay nicely attached and you can perform more assays, unlike from the 2D surface, when the cells after 7 or 14 days of experiment are too dead, actually wash out the monolayer of the cells, perform more assays, more washing of the cells. You have to be really careful. I would like to also share some tips and tricks, which you also can find on our websites, Inocure CZ, in Department of Support, when we have some nice videos how you can detach the cells from the membranes, how you can stain the cells and perform some assays, also seed the cells on the membranes. And also please hesitate to contact us on our emails or phone number find on our website. We will be always happy to help you consult your protocols and find the best option for you. This is, for example, one of the trick, because how I mentioned, the cells are in growth into the membrane. So to release the molecules from the cells or the color from the cells, from the metabolism of the cells, it's, for example, very handy to use homogenizer to release, for example, the cells, etc. So all these tips and tricks, yeah, we also learned by using the membranes and be very happy to share it with you. So thank you very much for your attention, and I really believe in future and necessity of the 3D to have proper results from in vitro tests, and which will get much more objective results for, let's say, even clinical use in the future. So thank you very much for your attention. If you have any questions, please, I'll be happy to answer them. You can share them in the chat.
Thanks, Eva. And thank you, Vana, for the nice and interesting presentation, which gives us more confidence, I think, to try cell culturing in 3 dimensions. In Germany and Austria, you can contact Biozyme for information or quotes. Check our website, biozyme.com, or contact your local Biozyme sales account manager. Now we are open for your questions. You can type them into the chat box, and we will answer them. We start with, are the InoMatrix 3D cell culture plates sterile when they are delivered?
Oh, thank you for the question. Yes, the InoMatrix plates are sterile. We sterilize them usually with ethylene oxide, but it can be sterilized on your needs, such as gamma irradiation plasma. However, the gamma irradiation, we could see some like it can influence properties of the membrane. So we highly recommend the ethylene oxide. And if you, for example, need to re-sterilize your plate, it's also possible to sterilize them with ethanol for 30 minutes and then properly wash by PBS, and then you can use the membranes.
Yeah, so I would like to also add very shortly that this is something why our product is actually very, very special for the customers, because it's coming ready to use. So actually, you don't need to add anything there, or you don't need to pipette. It's very easy to manipulate, especially in comparison with the, for example, hydrogels, where manipulation is quite tricky and a little bit complicated. This is solid, so it's very easy. You are just like the scaffolds are already attached there, so you are just adding your cells and you can start research activities.
Okay, thank you. Next one is, does InoMatrix 3D cell culture plates needs a pre-treatment prior to seeding the cells to enhance the cell attachment?
Yeah, thank you for the question. It's again, it depends on your protocols. We recommend to use your protocols same like for 2D cell culture, so you can definitely coat the fibers. Either we already have tried a coating with collagen or laminin 5. Also with uncoated fibers, and yeah, again, the more it's very suitable for this pilot test to find the optimal conditions for your cells. So it's yeah, depends on it's possible to coat the fibers.
Yes, but like for the basic experiments, for the like first trials and first experiments, there is no need any pre-treatment. So for it, yeah, as my colleague Eva said, it depends on application and on the type of cells. But for basic experiments, which are usually starting with, there is no need any pre-treatment.
Okay, thank you. And I would say we have maybe time for one last question. It came now up, the question, can the plates be combined with hydrogel? So
Yeah, that's a very good question. That's why we actually don't see hydrogels as like competition, because we are happy still if 3D cell culture environment is built. So one of the biggest advantages of our product from the point of sales and marketing is that it's actually able to combine with hydrogels, which is perfect because many people started with hydrogels, especially with Matrigel. But there is several problems. It's for longer discussion. But we have many clients coming to us with this question, and they can actually use it in combination with hydrogels. So it's very good for them because they can solve some issues which they used to have with using only hydrogel. So the answer is yes, it's possible to combine, and it's very popular to combine it.
Okay. Thank you very much for this very, very interesting talk again. And other questions will be answered later via email. And as you mentioned, if the customer is outside Germany and Austria, you can just contact Inocure directly. If you're working in Germany or Austria, please feel free to contact BioSime. And yeah, I think we wish you a very nice day, and thank you again.
Yeah, thank you very much for having us, and definitely visit BioSim website and also our website for more information, and then contact BioSim. So we are happy for cooperation and have many inspirative talks today.
Okay.
Thank you.
Bye.
Bye-bye.
Thank you. Bye-bye.